Namaskar. Today we will study the non-specific response in immunity. So there are three lines of defense in immunity, and what is immunity? Immunity is the overall defense mechanism of an individual organism against any invading agent or pathogen. So this invading agent or pathogen is called antigen, and in response to the antigen our body develops antibodies. So if the antibodies eliminate the antigen, we are free from the disease, otherwise we suffer from the disease. So there are three lines of defense. So initial defense will be the body parts like skin, mucous membrane, body fluids like tear, saliva, urine, which provides the physical or surface barrier. If it is not sufficient, then we develop the non-specific response, and these two together provides us the natural or innate immunity. And so immunity can be either innate immunity or acquired immunity. So when non-specific response is not sufficient, we develop specific response through acquired immunity, which can either be naturally acquired or artificially acquired. Then the non-specific response includes vascular events and cellular events. So when the blood vessel is involved, we have vascular events, and there will be vasodilatation, and there is increased vascular permeability because of all these autocoids like histamine, prostaglandin, causing vasodilatation, histamine, platelet activating factor, cytokines, nitric oxide, increasing vascular permeability. So because of this, there will be accumulation of fluid exudate, which contains 4 proteolytic enzyme cascades: the complement system, the coagulation system, the fibrinolytic system, and the kinin system. So the coagulation system, fibrinolytic system, and kinin system support the complement system, and the complement system is the main system which is involved in eliminating the foreign pathogen or the bacteria. And you can see, you can just again discuss this coagulation cascade. We have extrinsic pathway and intrinsic pathway. When there is tissue damage, the tissue factor is released. This is extrinsic pathway. And intrinsic pathway, when there is damaged blood vessel, the factor 12 or contact factor. In vitro, it is also called glass factor. In glass tube also this can be activated. So the step 1 is formation of the prothrombinase, and this prothrombinase converts prothrombin to thrombin. So in first there will be activation of the factor 10, which along with factor 5 and calcium forms the prothrombinase. Then this prothrombinase converts prothrombin to thrombin, so activation of factor 2 occurs. Then this thrombin activates the fibrinogen or factor 1. Initially there will be loose threads, and this thrombin also activates the factor 13, which causes the stabilization of fibrin loose threads into solid threads or clot. So this is coagulation cascade. And so fibrinolytic cascade, the insoluble fibrin is converted to soluble fibrin. There is dissolving of clot in presence of plasmin, which is the active form of plasminogen. And then the kinin cascade, kininogen is converted to bradykinin in presence of kallikrein. Bradykinin is a nonopeptide which is then inactivated by kininase 1 and kininase 2. Kininase 2 is nothing but the angiotensin converting enzyme. So all these three cascades, that is coagulation cascade, the thrombin, fibrinolytic cascade, the fibrin, kinin cascade, the bradykinin, they support the classical pathway in case of complement cascade. So complement cascade consists of 11 complements, C1 to C9, with 3 subforms of C1. So 11 complements are there, and the most important complement is the C3. And the C3 is activated to C3A and C3B, by two pathway, classical pathway and the alternative pathway. So classical pathway, this conversion is activated by C1, C4, C2 and the products of the coagulation cascade, fibrinolytic cascade and kinin cascade. Alternative pathway, it is activated by microbial products, endotoxins or polysaccharides on the bacterial cell surface. And the C3A acts as an anaphylatoxin, and it also releases histamine, and it is a spasmogen, and by releasing histamine it increases the vascular permeability. So C3A is an anaphylatoxin. C3B is an opsonin. Opsonin means it will attach to the surface of the microorganism and facilitate the phagocytosis, facilitate ingestion by macrophages. Then so it binds to the surface of the microorganism and helps the immune system. That means by facilitating the ingestion of those microorganisms by the monocytes or macrophages. Then C3B converts C5 to C5A, and C5A is a potent chemotactic factor. We call it as a chemotaxin. That means it attracts the phagocytes, the neutrophils, to the site of microbe invasion, and due to increased vascular permeability, these neutrophils easily penetrate those areas, and those neutrophils then cause the phagocytosis of the microorganism. Opsonin also forms this complex of C5, C6, C7, C8, C9, and this complex is called membrane attack complex, MAC, which causes the lysis of bacteria. So in this way, this complement cascade helps the immune system either through C3A or through C5A or through C3B or through this membrane attack complex, and helps in eliminating the pathogen or the bacteria. Then the cellular events, the blood cells are involved, and other than blood cells are also involved. So we can see which blood cells are involved. Except the erythrocytes, all other blood cells are involved, and multipotent hematopoietic stem cell is differentiated into myeloid stem cell and lymphoid stem cell. Myeloid stem cell develops megakaryoblast, proerythroblast, myeloblast, monoblast, and proerythroblast converts is differentiated into the RBC or erythrocyte, and this erythrocyte is not involved in our immune system. The rest, the megakaryoblast develops into platelets. Then myeloblast is differentiated into eosinophils, basophils, and neutrophils. Basophils are converted to mast cells, which are called specialized basophils. Then monoblast is differentiated into monocytes, and monocytes develop into macrophages and dendritic cells. Then lymphoid stem cell develops into lymphoblast, and this lymphoblast is differentiated into either large granular lymphocytes or small lymphocytes. The small lymphocytes are differentiated into T lymphocyte or B lymphocyte. There are two types of T lymphocyte: T helper cells, cytotoxic T cells. Then B lymphocytes are differentiated into either plasma B cell or memory B cell. The large granular lymphocytes are differentiated into natural killer cells. So all these have role in the immune system, in the cellular events of non-specific response. And we can see here the platelets release thromboxane A2, platelet activating factor, causing platelet aggregation. Then the morphonuclear cells or granulocytes like neutrophils, eosinophils, and basophils. Neutrophils release myeloperoxidase and defensins, and neutrophils cause the phagocytosis of the microorganism, then because of these enzymes it causes the destruction. So phagocytosis followed by destruction of the invading bacteria is caused by neutrophils. Eosinophils release proteins like eosinophil major basic protein, eosinophilic cationic protein, and neurotoxins which kills the parasite and cause allergic reaction. Basophils are developed to specialized basophil cells or mast cells which release histamine, 5-hydroxytryptamine, leukotrienes, which cause anaphylaxis mediated by immunoglobulin E. So they produce immunoglobulin E mediated anaphylaxis. The monocytes or macrophages, they release MCP1 or monocyte chemoattractant protein 1, or they produce the regulated on activation normal T cell expressed and secreted (RANTES), regulated on activation normal T cell expressed and secreted (RANTES), and they help in your opsonization. And some of the macrophages are converted to your dendritic cells, and they act as antigen presenting cells. That means they present the antigen so that the T lymphocytes can act on them. Then the tissue cells, that means cells other than the blood, we can say the vascular endothelial cells, which release nitric oxide, prostaglandins, endothelin, platelet activating factor, plasminogen activator, cytokines. All these cause vasodilatation and increase the vascular permeability. Neurons release 5-hydroxytryptamine, kinin, neuropeptides, all of them having role in Then mast cells, these are the specialized basophils releasing histamine, 5-HT and leukotrienes. Tissue macrophages, again the monocytes are developed into macrophages. Then the natural killer cells, some of the large granular lymphocytes are developed to natural killer cells which kill the invading microbes. Then some of the monocytes are differentiated into dendritic cells because they have the structure like dendrites. The dendron in neurons, similar structure is there, so because of that these cells are called dendritic cells. And these dendritic cells again act as the antigen presenting cell. Antigen presenting cell means when the antigen comes, the antigen enters into the cell, and then it is processed and presented on its surface in the form of major histocompatibility complex. So those cells are called antigen presenting cells because they present the antigen and they are expressed through major histocompatibility complex. Then the T lymphocyte on its surface expresses CD or CD receptor or cluster of differentiation, and these CD recognize the major histocompatibility complex and then they process to develop the specific immune response. So these are all tissue cells like vascular endothelial cells, neurons, mast cells, macrophages, natural killer cells, dendritic cells. So the blood cells as well as other tissue cells are involved in the cellular events. So this is all about the non-specific response, and the specific response we will discuss in the next video. Thank you.